So the first phase of karyokinesis is prophase. During interphase, the chromosome number become double, but the chromosomes, they are not visible. Even if we use electron microscope, the chromosomes are not visible, but we can observe chromosomes using histological strains. The stain which is used to observe chromosome is Fulgen stain. For nucleus we use hematoxylin stain, and for cytoplasm we use eosin stain. So this network of chromosomes which is observed using this stain is known as chromatin material. Now in prophase, the chromatin material, it becomes condensed or it becomes thick. By the process of folding, it becomes thick and thick. The chromosomes become more and more thick. Ultimately, each chromosome is visible, having two sister chromatids like this, which are attached at a centromere. Towards the end of prophase, now this whole condensation is occurring at the start of prophase. As soon as the end comes, the nuclear envelope disappears. Now the nuclear membrane will start disappearing. And the whole nuclear material, obviously, it will come in the cytoplasm. The nucleoli will disappear, and the mitotic apparatus, which I told you is composed of ester and spindle, it will be organized, and cytoplasm will become viscous. Now if we see it with the help of a diagram, the nuclear membrane has disappeared. These are the centrioles. The spindle fibers or the mitotic apparatus is organizing itself. The chromosomes, they are condensed, and we can easily see the sister chromatids. So this is what happens in prophase. Now let us talk about the next phase, which is metaphase. The metaphase is the next phase after prophase. At metaphase, the chromosomes are now duplicated structure, which consist of two sister chromatids which are attached at the central point, centromere, and the other name of centromere is primary constriction, as I have told you in the previous videos related to DNA and chromosomes. Now this centromere has a special area. Which is known as kinetochore. This kinetochore area has specific base arrangements. And proteins. So this is the point where the spindle fibers will attach. Now the kinetochore fibers they will attach at the kinetochore region of the chromosome, and they will align the chromosome at the equator of the spindle. That equator is known as the equatorial plate, and the other name is also the metaphase plate. Now if we see the diagrammatic view, now here the spindle fibers will arrange the chromosomes at the equator of the cell. Now each chromosome will receive one fiber from each pole. This is spindle fiber, and the fibers are attached at centromere at the point kinetochore. So this area is metaphase plate. Now let us talk about anaphase and telophase. Ana means to separate. It means at this stage the chromosomes will be separated, or you can say the sister chromatids will be separated from each other. Now this is the most critical phase of mitosis, and it has to ensure the equal distribution of chromosomes. The kinetochore fibers they will contract towards the poles. Now the stretching will take place, due to which the cell becomes elongated. The cell will stretch itself, the spindle fibers will stretch the cells, and the sister chromatids will be separated. And they will move towards the opposite pole. At the same time, the polar microtubules, they will elongate, and they will exert more force. The polar microtubules, they will elongate the cell, and they will help in the whole separation of the sister chromatids from the centromere. So as a result, half sister chromatid will be moving towards each pole. The last phase of karyokinesis is telophase. Now reaching of the chromosomes at opposite pole will terminate the process of anaphase, and it will start the telophase. The chromosomes will decondense. They will again start to become less visible. The chromosomes will decondense due to the process of unfolding. They became condensed by the process of folding, and now they are becoming decondensed with the process of unfolding, and ultimately they will disappear. Now the nuclear membrane will reappear. Now everything which was disappeared at prophase, it will start to reappear, and everything which appeared at prophase, it will disappear in the telophase. It's just the opposite. The mitotic apparatus will be disorganized, and ultimately the nucleoli which were removed, or you can say they've disappeared in prophase, they will also reappear. Mitotic apparatus will be disorganized. It will result in two nuclei, because this is separated. Now this is the structure of the cytoplasm, and we have two nuclear membranes which are forming around the separations, the nucleoli in each nucleus. And now the mitotic apparatus is disorganized. So this is telophase. Here the karyokinesis is completed because we can see clearly that one nucleus is divided into two nucleoli. Two nuclei are formed. These are the daughter nuclei. Now let us see how the cytoplasm divides the whole cell into two daughter cells, because here only karyokinesis is complete, still the cell division remains. So cell has to divide into two. That will happen with the help of cytokinesis, and that is the point where the cell division is different in plants and in animals.